In the ever-expanding landscape of gut immunology, IL-22 has emerged as something of a molecular unicorn, rare, potent, and almost too good to be true. Produced by a select group of immune cells, primarily ILC3S, Th22, and DT cells, IL-22 plays a dual role as protector and repairman. It doesn't attack pathogens directly. Instead, it empowers the gut epithelium to do so. It strengthens tight junctions, stimulates mucus production, promotes antimicrobial peptide expression, and drives epithelial cell regeneration. It's essentially a growth factor with a badge. And that makes it a tantalizing target for treating inflammatory bowel diseases, IBD, like Crohn's and ulcerative colitis, as well as diet-induced enteropathies. So, what does a future built on IL-22 therapy look like? First, there's the obvious approach. Give patients more IL-22. Recombinant IL-22 proteins, engineered, injectable versions, have already entered early-stage clinical trials. One of the most well-known candidates, F652, is a fusion protein that combines IL-22 with the Fc region of an antibody to extend its half-life in the body. Preliminary data from studies in liver disease and graft-versus-host disease show that it's safe and promotes epithelial regeneration. Its application to gut inflammation is now under active exploration. The idea is to deliver IL-22 during flares or early signs of mucosal erosion, giving the gut lining a regenerative boost without broadly suppressing the immune system like steroids or TNF blockers do. But simply adding IL-22 isn't always straightforward. Too much can be a problem. There's evidence that excessive IL-22 can fuel tumor growth in tissues where epithelial proliferation gets out of control. This makes precision timing and delivery a top concern. Smart delivery platforms like gut-targeted nanoparticles, microencapsulated oral formulations, or even genetically modified probiotic bacteria that release IL-22 on-site are being developed to reduce off-target risks. Another strategy is to enhance the body's own production of IL-22. This is a more subtle and elegant intervention, and potentially longer-lasting. Several compounds are being investigated for their ability to activate IL-22, producing cells, particularly ILC3S and Th22 cells. For example, agonists of the aryl hydrocarbon receptor, AHR, a transcription factor activated by dietary polyphenol and certain microbial metabolites, are known to increase IL-22 expression. Synthetic or plant-derived AHR ligands could be used therapeutically to boost endogenous IL-22 levels in a more controlled fashion, mimicking how certain foods or microbes naturally stimulate mucosal immunity. Cytokine synergy is another route. IL-23 and IL-1S are both upstream activators of IL-22 production in innate lymphoid cells. In a twist of irony, IL-23 is already a therapeutic target in IBD, where it's blocked, but some researchers are exploring whether low-dose stimulation could be selectively used to coax IL-22 expression during remission phases. The trick, again, is finding a Goldilocks zone, just enough to trigger repair without fueling excess inflammation. Then there's the idea of using IL-22 mimetics, small molecules or peptides that activate the IL-22 receptor, IL-22R1/IL-10R2 heterodimer, without requiring the cytokine itself. This approach is still in the conceptual stage, but receptor-specific agonists could theoretically bypass the challenges of protein stability and immune recognition. These drugs would act like a biochemical decoy, flipping on the gut's repair machinery without waking up the broader immune system. Another intriguing area is gene therapy or engineered immune cell infusion. Adoptive transfer of IL-22 producing cells, either autologous or from allogeneic sources, is being studied in preclinical models. For example, harvesting ILC3 precursors, expanding them in vitro, and reintroducing them into the gut could help reboot local mucosal immunity in patients with chronic epithelial failure. With advancements in CRISPR, Cas9, and synthetic biology, the idea of engineering smart immune cells that secrete IL-22 only under specific conditions, e.g., in the presence of gut dysbiosis, is no longer sci-fi. Finally, we shouldn't overlook the role of diet and microbiome-based interventions. Certain dietary fibers, fermented foods, and polyphenol-rich compounds, like those from berries, green tea, and cruciferous vegetables, are known to activate AHR and stimulate IL-22 through microbiota-dependent pathways. Microbial consortia that encourage butyrate and indole-3 aldehyde production, metabolites known to enhance IL-22, could form the basis of next-generation probiotics or synbiotics. The idea isn't just to fix the gut with drugs, but to train it with the right microbial cues and nutrients to self-regulate. Of course, none of these approaches come without caveats. IL-22's effects are context-dependent. In a healthy gut, it's a guardian. In a dysplastic one, it might be an enabler of excessive proliferation. It's not an anti-inflammatory agent in the classic sense. It's a tissue repair hormone with a temperament. But if researchers can harness its regenerative powers while dodging its darker tendencies, IL-22-based therapies could revolutionize the treatment of chronic gut inflammation, offering precision without suppression, repair without wreckage, and relief without relapse. So, in a future where high-fat diets, stress, and microbial insults have left our gut lining frayed and dysfunctional, IL-22 might just be the scaffolding we need to rebuild the fortress, one cytokine molecule at a time.